Hello, my dear students. Today we are going to discuss about one of the important topics being asked in CBSE exam many times, pollen pistil interaction. So pollination, it does not always guarantee you fertilization. The pistil can recognize whether pollen is compatible or incompatible through chemical signals and then either accepts or rejects it. You know that there are some chemicals released through by these synergids and antipodals. So that helps in recognizing the pollen grain, whether the pollen grain is compatible pollen grain or it is incompatible pollen grain. Now, after successful recognition, if it recognizes the pollen grain, then pollen germinates and forms a pollen tube. You can see in this diagram over here, it has been shown, this diagram, this one. This is the pollen tube germination is happening here. And then this is pollen tube over here. Pollen tube germination is happening and it is taking these two male gametes towards the embryo sac where you have the egg apparatus. So this thing over here has been shown how the pollen grain is growing, germinating on the stigma, both of these things. Then the tube grows through the stigma, style, ovary, and finally enters the ovule through the micropyle end. So this is your micropyle end over here in the ovule, so it will be entering through this micropyle end. It is guided by the apparatus that is there in synergids, that's called filiform apparatus. You know that we have discussed that thing. Then in plants shedding two-celled pollens, the generative cell divides during the pollen tube growth. We talked about 60% of the pollen grains, they are shed at two-cell stage and 40% at three-cell stage. So what is happening here in plants shedding two-celled pollens, the generative cell divides during the pollen tube growth. So here when the pollen tube growth happens, during that time in the pollen tube itself, the generative cell divides into two. So in plants shedding three-celled pollens, both male and gametes are present from the start itself. Just remember these 2 points, they're very important. When we talk about this, you can see here on the slide, pollen germination on the stigma, pollen tube formation via germ pore. Germ pore, where you don't have sporopollen, and then tube grows through the stigma and style, reaches to ovary, enters ovule via micropyle, enters synergids through the filiform apparatus. All these things I have already taught you. Now, what is mentioned in these diagrams is important. As I told you, this diagram, the first one here, and this has shown how the pollen grains are germinating on the stigma. And then second one also, this is the third one where it has shown that pollen tube is developing because the pollen grain was compatible with the stigma, and through the chemical interaction the pollen tube is developed and the two these male gametes, they reach to the egg apparatus. Then in this diagram D1, it's an enlarged view of an egg apparatus showing entry of pollen tube into the synergid. You can see here, this is the pollen tube, and it is entering into the synergids through the guidance of the filiform apparatus. Then this one, E, discharge of the male gametes into the synergid and movements of the sperms, one into the egg and other into the central cell. You can see here, this has been shown properly in these diagrams. So these diagrams can be asked in the exam. They can be given in your question paper itself. You have to recognize what is happening in this process. So these can be asked in the exam. Just remember that. I hope you like this video. Thank you very much.